Welcome to part 1 of Immunology. You may have noticed, as indicated by 2.5 weeks worth of it, immunology is a massively important topic. We will keep coming back to specific aspects of it, specific mechanisms in terms of pathogenesis, and just about any condition you can think of involves a degree of an immune process. Even things like aging. The way we age is dominated by cytokines, inflammatory mediators. It is a huge topic. We're going to try and cover the basics, and in this first part we'll look at physical barriers, but then we'll take it on to the innate response. The innate response is common to all organisms. It's nothing special that we have. When we get to the adaptive response, that's the clever stuff. The innate response, however, is quite complex, and in order to understand it, we've divided it into the cellular component, so in other words, the leukocytes, the white cells, and also the humoral component, which are the chemicals. We have no way covered anywhere near all of the chemicals, and you might meet more when we get into clinical reasoning, but it's a good basis to start from. So we'll talk about the innate response, but also a key part of this, and it's almost inseparable from inflammation. The inflammatory processes really are the innate immune processes, and we will look at that again in terms of the cellular and the humoral component, but then we'll try and put it together into a sequence of events with those chemicals and cells all coming into that. So it's quite a complex topic, but I really can't emphasize enough how important it is in terms of disease, in terms of pathogenesis, or in terms of outcomes of other pathogenic mechanisms. So let's talk about the levels of defense. We have 3 levels. The most basic really is the physical barriers. These are the barriers that prevent invasion by a pathogen. It's immediate because it's always there. It's also non-specific. And what we mean by non-specific in this context is it doesn't have the ability to recognize a pathogen. It can't even recognize a bacterium from a virion. And then if that fails, we get into the innate immune response. Again, this is non-specific. We do have methods of recognizing pathogens and binding to either pathogens or to damaged host tissues. But it has no immunological memory. In other words, once it's dealt with the invasion, it can't remember ever meeting that pathogen before. Therefore, the response is always going to be at the same speed and the same strength. But it has some recognition. It can recognize damaged tissue from pathogen. It can also significantly recognize self from non-self. In other words, it can recognize pathogen from our own cells. That then leads in through something called an antigen presenting cell that we'll eventually talk about, to the adaptive immune response. Now, this is something that is unique to more complex beings. So an earthworm doesn't have an adaptive immune response. It is mobilized, as we said, via the innate system through this antigen presenting cell. Its response is pathogen, or more importantly, antigen specific. So once activated, we will have long-term immunological memory to an antigen, and will from there on have the ability to resist that antigen very quickly and in a very strong way. So first exposure creates this immunological memory to cope with any subsequent challenges by that specific pathogen or antigen. So let's have a look at a bit more terminology. Microbe. A microbe is the contraction of the word microorganism, really. It's a microscopic organism, and this includes bacteria, viruses, fungi, or protozoa. Microorganism, as I said, really it's more or less microbe, but some definitions don't include viruses because viruses are not independent organisms. They can't replicate independently. Bacterium, I'm sure we all know this one, a single bacterial microorganism. And virion. Virion is a complete encapsulated virus. A prion, this is the last time really we're going to mention prions, but this is an infectious protein particle. So it's similar to a virus in that it's not really a functional cell, but unlike virions, it doesn't have nucleic acid. So an example of this might be Creutzfeldt-Jakob disease. All we've got is a collection of proteins. Pathogen. So a pathogen is a microbe, but it's a microbe that causes pathology. It causes disease or illness, and they are not part of the normal commensal microbiota that we have. They are pathogenic. Usually a few pathogens can be controlled, and we come in contact with them all the time. The difference is if they're allowed to colonize, if they're allowed to replicate, and many of them replicate very, very rapidly, and then they release toxins, they cause cellular damage, they cause all sorts of problems, stimulate an acute immune response, and ergo we get the signs of illness. So they are pathogenic. Antigen relates to any substance capable of inducing an immune response and reacting with the products of that response. So an antigen could be an allergen, or it could be any part of a pathogen, or any part really of a damaged host cell. It's something that stimulates an immune response. So it can be exogenous, in other words coming from outside the body, endogenous, coming from within the body, or we could be talking about autoantigens, as in autoimmune conditions. Epitope is really getting a little bit smaller now. So here we're talking about just a few amino acids from the antigen to which antibodies will bind. So an antigen can have multiple epitopes that will stimulate an immune response. Lots of little parts of that antigen could stimulate, could bind to antibodies, could bind to T-cell receptors, and could stimulate an immune response. So a single antigen may have many epitopes. Therefore, many epitope-specific antibodies can bind to different epitopes on a single antigen. So, for example, we could have multiple antibodies that will respond to an antigen, but to different parts of it. So that was our terminology so far. So let's have a look at those layers. And if you remember, the first layer that we talked about were the physical barriers. These are the things that are there all the time. They're non-specific. They are designed to stop anything entering. And of course, the first one that springs to mind is the skin. The skin, we have an epidermis and a dermis. In the epidermis, if you remember, the epithelial cells mature as they get towards the top. They become keratinized and then they slough off. Remember the little desmosomes all breaking together. So individual cells slough off constantly from the surface, causing lots of dust in our houses. But they're also taking any potentially colonizing microbes, pathogens, with them. The other thing that the skin has is the acid mantle. This is produced from sebum. It has a pH of 5.5, and this is why it used to be that a lot of soaps would say, oh yes, pH 5.5, that's, you know, not damaging to the skin. And they were basically quite right. That pH of 5.5 inhibits pathogens from colonizing and also protects against alkalines. So these days, of course, we have lots of antibacterial soaps which are great for getting rid of these pathogens, but not really so good for our skin. The other thing is that the follicles, the hair follicles and the sweat glands in moist areas secrete lysozymes, the enzymes, and toxic lipids. These help to destroy any colonizing pathogens or prevent them from colonizing. The saltiness of sweat also inhibits bacteria. So bacteria are a bit like slugs, they don't like salt. Nasal hairs. Nasal hairs prevent entry of any pathogens via the nose. So again, it's quite a mechanical, physical barrier in that we've got this wall of hairs to try and trap them all from entering. The other thing is that the skin is generally cooler than body temperature, and this inhibits pathogen replication. The mucosae are an extension of the skin. If you remember when we talked about covering and lining epithelium, we talked about the mucous membranes extending inwards, lining all the organs that are open to the inside, and they're an extension of the skin. So we have similar mechanisms plus a few extra. So they line the cavities that are open to the exterior. They also slough cells off the surface, like the epidermis does. In addition, though, we have mucus. Mucus traps microbes and foreign material. And mucus, mucins, the solid component of mucus, have pattern recognition receptors that can actually bind and hold any invading pathogens, which of course then get removed with the mucus. So the mucus itself works together with the cilia, and we have this mucociliary clearance. The cilia waft the mucus up to the outside, and we get rid of it. The mucosa and the mucus also contains lysozymes, lactoferrin and lactoperoxidase. These are all chemicals that help to destroy any colonizing pathogens. The other thing that both the skin and the mucosa have are resident microbiota. These are our, you could say, friendly bacteria, but we're not just talking about bacteria here. This are our commensal microbes. The skin and mucosa also have Langerhans cells. These are a type of white cell, leukocyte, known as a dendritic cell, but they have adapted slightly and they tend to be more skin and mucosa specific. So they are innate immunity, skin specific dendritic cells. And again, we have salt and malt. We have this lymphoid tissue just below the basement membrane. We have skin associated lymphoid tissue, and we have mucosa associated lymphoid tissue, salt and malt. These two areas of lymphoid tissue are a bit like if you took a lymph node and flattened it out and stuck it just below the skin and the mucosa. So we have lymphocytes very, very close to these physical barriers. Those lymphocytes are the intraepithelial T and B lymphocytes from the adaptive immune system. And we also have mechanical removal. So we've talked about the mucociliary clearance. We've talked about mucus being moved, wafted by the cilia out of the body with any trapped microorganisms or contaminants or dust, anything like that, back up the throat for expectoration, or probably slightly cleaner, swallowing. Of course, when we swallow them, they get into the gastric acid and they're destroyed. We also have the cough and sneeze reflex. That's a way of removing mucus with these trapped microbes. Vomiting and diarrhea is also flushing out any invading pathogens. And other methods of flushing that we might not think of quite so obviously are things like tears, saliva, sweat, and blood from a skin break. And if you think about it, if you have a needle stick injury, the first thing you're told to do is to make it bleed. Squeeze it. Squeeze the blood out, because with that you're flushing out any potential pathogens that have invaded through that skin break. Back to our microbiota. How these work is that we have antagonism by this normal flora. Microbiota is a more accurate and contemporary term. So this includes 100 trillion microorganisms, and we're mainly talking about bacteria and yeasts in this case. They don't usually cause any harm to us as long as they're in the right place. That's quite important in terms of them then getting in the wrong place and causing pathogenesis, causing illness. In some cases they actually benefit, particularly in the GI tract, because here we depend on some of these microorganisms to break down ingested solutes so that we can absorb them. So the microbiota is found on skin, in saliva, on the mucosae, conjunctiva, and in the intestine, in particular the ileum. They inhibit the growth of many of the pathogens, and they do this by competing. They're competing for space, but they're also competing for nutrients, nutrients that the pathogens would otherwise use so that they can replicate. Or in some cases they secrete toxins that actually inhibit pathogen replication, inhibit colonization. So those were our physical barriers. Those are there all the time. If they fail, our next layer of defense is the innate immune response. And as our little pink dinosaur, not so sure about pink, but as our dinosaur there demonstrates, it's not very specific. So it will attack, but it needs to be told what to attack. So the innate response involves the recognition of self from non-self. In other words, it needs to recognize healthy host cells, our cells, from any invading pathogens or from any damaged or dying cells. It also heavily involves the inflammatory response. So this is all about really inflammation. And inflammation is generally in an acute inflammation a progressive process. It starts with vasoactive changes that we'll talk about and leukocytosis. Leukocytosis, of course, is when the white cell count goes up. We have more leukocytes released from the marrow into the blood. It also involves emigration. This is where our leukocytes need to get into the tissues. They need to get out of the blood and into the tissues. Once they're there, they need to know where to go, and this is chemotaxis. Once they get to the target, you remember phagocytosis from very early on when we talked about transport, and we talked about this as a form of endocytosis. So this is when our white cells will get into the target area and they will start phagocytosing. Once they've phagocytosed, some of those leukocytes can become antigen presenting cells. And this is our link then that takes us into the adaptive response. There are a few other innate immune response components that we'll talk about. There is fever. Fever is a protective response. We'll need to talk about natural killer cells because these are actually lymphocytes. So they're like the T and B cells, they're all lymphocytes. If you like, they're born in the adaptive system, but they operate in the innate system. And we'll also need to talk about antimicrobial peptides and proteins, or at least a couple of them. So what about this recognition of self from non-self? So in order to eliminate pathogens or damaged cells, of course, we must be able to recognize them. We must be able to distinguish them from all the healthy cells. So we need to have an idea of what is non-self. We need to have some markers that identify non-self. One of these is a generic term, pathogen-associated molecular patterns. So there are quite a few of these molecular patterns that are associated with pathogens. For short, we call them PAMPs. So they are molecules unique to groups of related microorganisms. So a key example is lipopolysaccharide. Bacteria, many bacteria on their cell wall carry these lipopolysaccharides that are recognizable to our defense mechanisms. Also maybe lipoproteins. It may be damaged or altered lipoproteins. It may be bacterial and viral DNA or RNA patterns Or, in the terms of yeast cells, it could be the glycolipids that yeast display. All of these will identify them as a pathogen. They are the molecular patterns associated with these pathogens that will identify them. And another one we've got to think about, because the immune response and inflammation is not all about infection. You really need to keep that in your mind, particularly as we move through into clinical reasoning. Because we also have damage-associated molecular patterns. So this is about our own cells. Our own cells may become stressed, injured, they may become infected, particularly by viruses, or they become transformed. And they then display damage-associated molecular patterns. So this includes something called heat shock proteins. Heat shock proteins are released to try and protect proteins from high temperatures. They try and stop proteins from denaturing, from falling apart. If they can't do that, they will then mark that cell as for destruction. You need to destroy this cell because it's falling apart and it's going to cause a problem. Or a DAMP might be altered membrane phospholipids. So any damage to a cell will alter those phospholipids in the outside plasma membrane, and that will get picked up as a damage-associated molecular pattern. Now, what recognises them, of course, it's all very well having these markers, but you need something to then bind to them to recognise them. And this is where we have pattern recognition receptors. So we have molecular patterns, whether they be pathogen associated or damage associated, and it's quite logical that we then have pattern recognition receptors, PRRs. So PRRs can then bind to PAMPs and DAMPs. If they're on a phagocyte, this can stimulate phagocytosis. So that pathogen, in the case of a pathogen-associated molecular pattern, or that damaged cell, as in a DAMP, will become phagocytosed. It will be got rid of. Whenever a phagocyte phagocytoses, what it also does is start secreting inflammatory mediators. Basically what it's doing is calling for help. It's releasing lots of different inflammatory mediators that will facilitate help coming to it. So, and it does this through this intracellular signaling. Nuclear factor kappa B is a key signaling protein in inflammation. Once that's activated, then it starts the whole process of transcription and translation of a lot of inflammatory mediators that are then secreted from that cell. Those inflammatory mediators will upregulate the immune response. So the minute you get one, say for example macrophage phagocytosing, it will then start a whole inflammatory cascade through the use of the chemicals of the inflammatory mediators. Pattern recognition receptors, they're found on macrophages. So a macrophage will bind via its pattern recognition receptor to a PAMP or a DAMP and will then phagocytose that target in the same way as dendritic cells do. In some cases, endothelial cells, they're not our professional phagocytes, but they can respond via pattern recognition receptors. And lymphocytes, our T and B cells, our natural killer cells, they have pattern recognition receptors as well. And if you remember, I said also in mucus, we have some pattern recognition receptors. Pattern recognition receptors, PRRs, enable these cells to bind to pathogens or to damage cells, depending whether it's a PAMP or a DAMP. The outcome is phagocytosis, and through that sequential secretion of inflammatory mediators through the activation of NFKB, the intracellular signaling molecule, that phagocyte will then release lots of pro-inflammatory chemicals to stimulate an immune response and basically call for help. So that was markers of non-self. Markers of self is all about reporting your status. It's a bit like a ship running up signalling flags in the old days when you had big sailing ships. Now, the flag post in this case is something called major histocompatibility complex, MHC. We all have these little proteins, these little flag posts, and we display them on all of our cells. They are unique to us, unless you have an identical twin. And they were actually discovered in the 1940s because, of course, they were treating a lot of people with burns. They were trying to do tissue grafts, and they couldn't understand why they were being rejected. And this is because each individual has their own unique MHC flagpole, if you like. If you read American texts, you may see HLA. HLA is MHC. They just insist on calling it something else. So we've got two classes of MHC. MHC1, this covers almost all nucleate cells. So you remember that glycocalyx, we've got all these glycoproteins sticking out of the cells like little satellite dishes. Some of those will be MHC1, up to 100,000 per cell. Now, generally, if that cell is a happy, healthy cell, that MHC1 that has, you can see there, a little notch in the top. In that little notch, it will have normal, healthy proteins, only around 10 amino acids. We're talking a really small protein. And this is demonstrating to any other cell that might attack it that, look, I've got healthy, normal proteins. I'm a healthy, normal cell. Please don't attack me. However, what happens is if that cell becomes infected, for example, with a virus, or it becomes damaged, it will then take either unhealthy amino acids, damaged amino acids, or maybe viral proteins, and change those MHC1 for ones that hold the antigen. They hold bits of damaged cell, bits of damaged protein, or bits of pathogen. This is now putting a flag up to say, hang on a minute, I'm not very well. Can you come and attack me, please? So we have, if you like, the MHC being our flagpole. We can have a healthy flag, or we can have an unhealthy flag. Depending on whether the flag's healthy or unhealthy, that is advertising to all the white cells, the leukocytes around, what they need to do about it. MHC2 is very similar. You see we've still got a notch for antigen, but this is found only in our macrophages, in B lymphocytes, and any cell that can become an antigen presenting cell. This is about antigen presentation of anything that maybe has been phagocytosed to lymphocytes. It's about running off to lymphocytes with this little flag that says, oh, look what I've just phagocytosed. This is a bit of a worry, isn't it? I need you to react. So MHC1, although our leukocytes will have MHC1 and MHC2, MHC2 is the only one that's used for antigen presentation to activate the adaptive response. Let's have a look at a diagram. So here we have a pattern recognition receptor, a PRR, on a phagocyte. So here we have two potential targets. We have a pathogen bearing a pathogen-associated molecular pattern. Those are the little triangles sticking out from it. And we have a damaged cell. That damaged cell, its little triangles, are damage-associated molecular patterns. Either of these will bind to our pattern recognition receptor. So here we go, damaged cell got there first. He's bound to the pattern recognition receptor. That's stimulated phagocytosis. He's taken into a phagosome, which will then fuse with a lysosome to become a phagolysosome. Another way of binding is through opsonization. We'll come back to opsonization when we talk about the specific proteins that do this. One of the things that can do this are antibodies. So the antibodies that in their monomer state, in their simplest form, are a Y shape, will bind to the target and they will opsonize it. They will mark it for destruction. In other words, for phagocytosis. On the phagocyte, in order to do that, we have another receptor. This is called an FC receptor, FCR. FC relates to the pointy end of our Y shape, the bit that's not bound to the target. So the FC end always binds to our own cells, while the other ends, two of them in that case, are called the FV ends. They bind to the target. So hence this is called an FC receptor. So our antibody can bind to the FC receptor while bound to the target. This is opsonization. And that will also stimulate phagocytosis. So we've got pattern recognition receptors for PAMPs and DAMPs. We've got an FC receptor for the FC end of an antibody that's bound to a target. And our other opsonin is complement, a complement protein called C3b. We'll talk more about that later. So here we've got a target to which this complement protein is bound in the same way as the antibody bound to its target. It has opsonized the target. On our phagocyte, we have a complement receptor. Guess what's going to bind to that? Yeah, the complement, while bound to its target. And that will stimulate phagocytosis. So we've got three possible ways of binding to a phagocyte there. Through a pattern recognition receptor, through an FC receptor, or through a complement receptor. All of these will result in phagocytosis. So these are all markers, if you like, of non-self. And of course, the phagocyte, as we said before, the minute it phagocytoses, NFKB is activated inside the phagocyte, And that stimulates the synthesis and the secretion of lots of inflammatory mediators, prostaglandins, TNF-alpha, interleukins, colony stimulating factors, lots of different chemicals that are going to stimulate an innate immune response. MHC, as we said, our phagocyte could have MHC1 or 2 because it's phagocyte. If it was a normal host cell, not a white cell, not a leukocyte, it would have MHC1. And a phagocyte could have either. In this case, our cell is displaying healthy proteins. I'm okay, Jack, go away, leave me alone, don't kill me. However, if it displays a little bit of viral RNA, for example, a few amino acids from a damaged organelle, then that is displaying an unhealthy flag. It's displaying a sign that this cell is damaged or infected. Or, in the case of a phagocyte, what it's actually doing is it's displaying something that is then going to go off and bind to a lymphocyte and activate some lymphocytes. If this was our phagocyte, that was MHC2 or MHC1, and it's displaying a little bit of something it phagocytosed, so there it phagocytosed our damaged cell, could break down a little bit of that, put that up in its flagpole, its MHC, and present that to lymphocytes to activate the adaptive response. So from there, that could then bind to a T cell, bind to a B cell, bind to all sorts of things, and that would then be activated, and off we go for a full-blown immune response. So you can't talk about innate immunity without talking about the inflammatory response. Inflammation is caused by cellular injury, and this cellular injury may be a result of multiple things, not just infection. So it may be due to trauma, may be due to a hypersensitivity reaction, that may be autoimmune, or it may be an allergic, atopic reaction. It may be due to chemical agents, infection via microorganisms, so bacteria, viruses, fungi, etc. Temperature extremes, so burns or cold burns, or any dead cells, whether they be host cells due to microorganisms and infection, or dead parasites. It's a non-specific defensive response, hence we're talking about it in terms of physiology. It's a normal defense mechanism in terms of acute inflammation. It forms part, a major part, of the innate immune response. And its role basically is to eliminate or restrict, sometimes by walling off, the initial cause of cellular injury. And then to eliminate any necrotic cells, so dead cells or dying cells, from the tissues or the injury site. So the classic phrase about inflammation is that it's damage limitation and cleanup in preparation for repair. The innate immune response in inflammation does, however, underpin many diseases and disorders, as I said, even aging. And this is where we start to get the switch over, really, from this acute defensive response into a more chronic response. If we keep stimulating inflammation, we will get chronic inflammation, and that becomes more damaging and more pathogenic. The innate immune response and the adaptive response, for that matter, both involve a cellular component and a humoral component. Humoral refers to chemicals. So we have cells and we have chemicals. The humoral part, what we're talking about are chemicals that are cell-derived, so they come from cells all over the body, and they are preformed, so they are synthesized in preparation, and then they're contained in the form of granules within specialist little vesicles within cells known as granulocytes. Site, of course, always refers to cell. These are cells with these granules of preformed chemicals. We then have cell-derived, newly synthesized chemicals. This includes things like prostaglandins and leukotrienes, platelet activating factor. These all come as part of the breakdown of phospholipids in the plasma membrane. Reactive oxygen species, which is actually ROS, which is a group of free radicals. Nitric oxide, TNF alpha, interleukins and chemokines, which are cytokines. They're a specific branch, if you like, of inflammatory mediators. And then in terms of chemicals, we have plasma derived, although really they should be explained as liver derived. They go around in the plasma in inactive form and then need to be activated, but they originally are synthesized by the liver. And this includes complement. We've already mentioned complement proteins as an opsonin in opsonization. It also includes kinins, and part of that cascade we get bradykinin, and it includes the coagulation factors. So we have cell-derived preformed, we have cell-derived newly synthesized, and we have plasma-derived chemicals. In terms of cells, all of these are white cells, all of these are leukocytes. So we can classify them as granulocytes that we just talked about. These are the cells that have these preformed granules of mediators, of chemicals within specialist little vesicles. Granulocytes include mast cells, neutrophils, eosinophils and basophils. In terms of cells, we can then have another classification of the professional phagocytes. Generally, the professional phagocytes are classed as dendritic cells and monocytes or macrophages. Monocytes are released from the bone marrow, and once they get into tissue, they become macrophages. Our third group here, or standing all on its own, are natural killer cells. Now, natural killer cells in the innate response operate in the principles of the innate response, but they are actually lymphocytes, so they have to stand on their own a bit. Lymphocytes is another group that operate within the adaptive response, not within the innate response. They have to be activated by the innate response. So let's talk about some chemicals. This is actually quite a small selection of chemicals, So we've picked out the main ones that you really need to know at this point. So we talked about cell-derived or plasma-derived. Cell-derived come from cells. Plasma-derived generally are synthesized in the liver, released into the circulation in an inactive form, and then have to go through various changes to become active inflammatory mediators. And as we said, cell-derived may be preformed and released as secretory granules, or they may be newly synthesized as required. The mediators, once they're released, are limited in their action, and it varies across the different chemicals. They're limited in their action by decay, by enzymatic inactivation, elimination, or inhibition. But we have to control them, otherwise we'd end up with an ongoing inflammation even though we've removed the trigger of that inflammation. So we have to get rid of them at some point. So preformed then, these are our ones in granules. We have histamine and serotonin. Histamine and serotonin are synthesized by mast cells, basophils, and platelets. Serotonin particularly in terms of platelets. They're then released by exocytosis or by degranulation of the granulocyte. Lysosomal enzymes, we know about lysosomes because these are what help break down the target when we get a phagolysosome in a phagocyte. But also they're the things in all cells that break down any proteins that we've finished with. Say we want to take down some channels, some receptors, and we want to break them down into their amino acids so they can be recycled. This is where a lysosome comes in. So lysosomes are a bit like the sort of stomach, if you like, of cells. They're little vesicles full of these lysosomal enzymes. Now these lysosomal enzymes, when released, can be quite damaging because they break down proteins, they break down collagen. And those enzymes can be released from neutrophils, from macrophages, and potentially really all cells when they break down. Newly synthesized, these are the ones that are stimulated to be synthesized and then secreted as and when they're required. This includes things like prostaglandins and leukotrienes, and they are synthesized via something called the arachidonic acid cascade that we'll talk about in a little while. It also includes cytokines. Cytokines is a group of newly synthesized inflammatory mediators that includes interleukins, and there's quite a lot of interleukins. There's about 30 odd interleukins. It also includes interferons, colony stimulating factors that might otherwise go by the name of leukocytosis inducing factors, which might give a hint as to their role. They're the ones that cause leukocytosis, the release of more leukocytes from the bone marrow. And it also includes, and this is really the big bad boy, tumor necrosis factor alpha, TNF alpha. And certainly in terms of chronic inflammation, TNF alpha is a very important cytokine, as are some of the interleukins. Newly synthesized includes platelet activating factor, and this is synthesized from phospholipids alongside really the arachidonic acid cascade, at the same time, because the arachidonic acid cascade that produces prostaglandins and leukotrienes by two different pathways uses phospholipids to do this. And reactive oxygen. Reactive oxygen species, ROS, is a group of free radicals. Free radicals have basically an unpaired valence shell electron, which makes them want to go off and bind with lots of other things. Now, normally in normal levels, that's great because they can act as signaling molecules. In large quantities, they can cause a bit of chaos. So these are free radicals, and they're activated by lysosome-based mediators. So the lysosomes themselves will help to activate them. They will convert some of their contents into reactive oxygens. Nitric oxide is very similar. It's produced from lysosomal action usually, and it's a very potent vasodilator. So potent that if you have a massive immune response, then it is released systemically and it can cause shock. It causes the drop in blood pressure that you see in sepsis. So it can be very significant. Luckily, it doesn't last very long. It tends to work locally because once released, it deteriorates very quickly. Which brings us on to our plasma-derived. As I said, these are synthesized in the liver and then they circulate in inactive form around the blood. So we have initially synthesized by the liver, we have factor 12, which is the Hageman factor. That is then activated and we can have two pathways from that. We've got to bog off again. So one of those pathways is the clotting system. The clotting system, we're talking about a whole sequence of activities, including the aggregation of platelets, the laying down of fibrin and clotting. We also have another pathway, originates with Hageman factor, and that's the kinin system. The kinin system, again, we have this whole cascade of chemical changes, this whole sequence, signaling sequence. But ultimately, what it produces that's important for us is bradykinin. Bradykinin plays a role in so many different ways in terms of the inflammatory response. Also synthesized by the liver, we have complement 1, C1. C1 circulates around in the blood in an inactive form. It can then be activated in various different ways. We'll talk about those a bit later, but once it's activated, we get several different complement proteins. Primarily the two C3s, C3a and C3b. We get C5a, and then we get C5b, C6, C7, C8 and C9. These all work together as something called membrane attack complex, and basically they put holes in cells. So, okay, let's go back to our preformed and have a closer look. These are synthesized as granules, stored in granulocytes in specialist vesicles. One of these that we mentioned was histamine. Now, histamine is really quite important in terms of the early inflammatory response. It is the main vasoactive amine. By vasoactive, we're including two actions here. One of them is vasodilation, not systemic, we're talking about local vasodilation, and also increased vasopermeability within that same local area. So it dilates vessels and it makes them leakier. Histamine also stimulates more mast cells to degranulate. So it upregulates its own action, if you like, by causing the release of more histamine. Now, whereas in vasodilation, if you think about the structure of a blood vessel, histamine binds to smooth muscle and causes it to relax, hence vasodilation. But actually it binds to different receptors in the smooth muscle of the bronchi and causes them to contract. So it causes bronchoconstriction. We also have heparin. Heparin comes from mast cells. Heparin stimulates bradykinin release from plasma proteins, and as we know, it inhibits clotting. And we talked a little bit about lysosomal enzymes. These can potentially come from all the cells in degranulation or in lysis. When those cells break down for whatever reason, the lysosomal enzymes will be released. And this includes lots of proteases, lots of reactive oxygens, lots of toxic chemicals. Lipases. Lipases break down fat, proteases break down proteins. So these enzymes, if they're released in large quantities, could potentially destroy healthy host tissue. Serotonin comes from mast cells and platelets, although most of it actually is produced in the GI tract. It has lots of different physiological and regulatory functions, and of course it also operates as a neurotransmitter when it's secreted from neurons. It's secreted constantly, not just in inflammation, but in inflammation it does play a key role. Which brings us on to our newly synthesized inflammatory mediators. So one of the main mechanisms of this is the arachidonic acid cascade. The arachidonic acid cascade produces something that we can categorize as eicosanoids. If you see eicosanoids, it refers to the products of this arachidonic acid cascade. The cascade itself is stimulated by either mechanical cell trauma, particularly trauma to the plasma membrane, including any variation in the plasma membrane, such as when phagocytosis occurs, or it can be stimulated by various other mediators or other eicosanoids, so it can stimulate itself, a bit like histamine, binding to extracellular receptors. So when it's activated, what happens is a phospholipase, in this case phospholipase A2, takes plasma membrane phospholipids and synthesizes arachidonic acid. And then arachidonic acid can produce eicosanoids by two different pathways that you're going to recognize. We have the COX pathway, cyclooxygenase pathway, so called because the enzyme cyclooxygenase converts the arachidonic acid to prostaglandins, prostacyclins, and or thromboxanes. And then we have the other pathway. The other pathway is often known as the LOX pathway, the lipooxygenase pathway. Again, so called because the enzyme lipooxygenase in this case, converts arachidonic acid into leukotrienes and or anti-inflammatory lipoxins. So we have various eicosanoids possible from either of these two pathways. They're not all secreted at the same time, otherwise we'd have a bit of a mess because they tend to do different things. But we do have these two separate pathways. We have two enzymes, cyclooxygenase and lipooxygenase, both converting arachidonic acid into different things. Now, eicosanoids are synthesized by all cells, all normal cells, apart from erythrocytes. They're usually the exception in so many cases. But not all cells synthesize the same eicosanoids, or metabolites of arachidonic acid. It depends on the enzymes present within that particular cell. So, for example, leukotrienes are generally synthesized by leukocytes, granulocytes, and epithelial cells. Thromboxane, as you might expect, is synthesized by platelets. Prostacyclin, which is a vasodilator, and it inhibits platelet aggregation, so opposite to thromboxane, is synthesized by vascular endothelial cells. Prostaglandin D2, PGD2, is the main prostaglandin from mast cells. So, and so on and so forth. Those are a few examples. So different cells have different enzymes that will result in different eicosanoids being produced. Eicosanoids rapidly decay or are destroyed by enzymes, which limits their action. Again, we said this is quite important in inflammation, because unless we keep triggering inflammation, we eventually want it to resolve. That's what makes it acute inflammation and makes it helpful. If it carried on happening, it will become damaging. So we need for these chemicals to degrade relatively quickly. So let's have a look at a diagram of the arachidonic acid cascade. You will see lots of diagrams on Google or in textbooks that are quite complex. So I've taken it and I've simplified it to the things you need to know. You will see diagrams with lots of other chemicals in between that really we don't need to talk about, certainly at this stage. What we do need to talk about is phospholipase A2. So remember when we were talking about signaling and we were talking about a G protein activating phospholipase C, in this case we have phospholipase A2, but we don't necessarily have a G protein activating it. It's an enzyme that is embedded in the plasma membrane. It uses phospholipids to make other things. In this case, what it makes is arachidonic acid. Along comes lipooxygenase, an enzyme, and it converts arachidonic acid into leukotrienes. So leukotrienes you'll see abbreviated to LT, and there's various of them. So you can have LTC4, LTD4, and etc., LTE4. Leukotrienes tend to cause vasoconstriction, so they contract the smooth muscle of blood vessels. They also cause bronchospasm and increased vasopermeability, and they're quite significant in terms of asthma. That's often where they're discussed. Now, lipoxins are generally anti-inflammatory. So we're not going to secrete leukotrienes and lipoxins from these cells at the same time because their effects are so different. There will at some point be the synthesis, there will be a change, and there will be the synthesis of an enzyme that will start producing lipoxins rather than leukotrienes. So lipoxins are anti-inflammatory. In our other pathway, in our COX pathway, we have the enzyme cyclooxygenase. Cyclooxygenase converts arachidonic acid into prostaglandins, so for example PGD2, or a really important one, PGE2. Now these cause vasodilation and increased vasopermeability. They also bind to nociceptors and stimulate pain, and as we'll see later on when we talk about fever, PGE2 is vital in the stimulus of fever. From the cyclooxygenase pathway we can also get prostacyclin and thromboxane. These have directly opposite effects on vessels and clotting. So we don't produce them both at the same time, it depends on the cell and it depends on the time. But these are all possible eicosanoids that result from the breakdown of arachidonic acid by either cyclooxygenase or lipooxygenase. And this, talking of asthma, this is why some asthmatics can't take NSAIDs, because of course NSAIDs work on the COX pathway. They block the COX pathway. So they reduce the potential for prostaglandins. Hence, we take them when we have pain or inflammation. But in asthmatics, a certain percentage of asthmatics, not all, but a certain percentage, what happens if you block the cyclooxygenase pathway is that the cells will put more effort into the lipooxygenase pathway. So you downregulate cyclooxygenase, then the cell will upregulate lipooxygenase. So of course those asthmatics, when they take NSAIDs, will get a higher secretion of leukotrienes, which with their effects of vasoconstriction, bronchospasm, etc., causes more problems in terms of their asthma. So let's talk about a couple of other newly synthesized cell-derived mediators. These are ones that aren't exactly a part of the arachidonic acid cascade. So platelet activating factor is generated from the phospholipids of the plasma membrane, but it happens after the activation of phospholipase A2. So it happens after the arachidonic acid cascade. Platelet activating factor upregulates most inflammatory processes and it induces platelet aggregation, so therefore it starts clotting. It's also a very potent vasodilator and bronchoconstrictor. So in other words, it relaxes vascular smooth muscle, but when it binds to receptors on bronchial smooth muscle, it causes contraction. Reactive oxygen species, we've talked about a little bit. These are molecules that contain oxygen, and they're so-called reactive because they have this unpaired valence shell electron. If you remember, the valence shell is the outer shell of electrons. Because it's unpaired, it makes them want to bind to other ions or molecules. It comes from oxygen that's been reduced. So in other words, that oxygen molecule has had an electron added to become more negative. They are a normal byproduct of oxygen metabolism during the electron transport chain, but a whole other set, if you like, in inflammation is synthesized via something called the NOx pathway by both neutrophils and macrophages to kill the phagocytosed bacteria. So, as I said, a normal level of this, it is used really as a normal signaling molecule, but then we make some extra via this NOx pathway, and that then starts getting busy with inflammatory processes and with breaking down any phagocytosed pathogens. Nitric oxide is known to be a major vasodilator. It's known for vasodilation. Its synthesis is stimulated by cytokines, so TNF-alpha, interleukins, etc., platelet activating factor, acetylcholine, and also, which is quite cool, shear stress on vessel walls. If you think about it, if your vessel has an obstruction like a clot, you get additional shear force on the vessel walls because the blood is trying to force through a narrowed gap. That shear force will then stimulate the synthesis and release of nitric oxide, which is a vasodilator, which helps you to overcome the obstruction in that local area. It's synthesized by endothelial cells and leukocytes via nitric oxide synthase. Now there's two types of this. There's eNOS, endothelial nitric oxide synthase, and there's iNOS, which is inducible. Don't worry too much about the difference between those two, or even remembering them particularly. Just so that you know that if you read a scientific article and it says that a particular drug or whatever increased the presence of eNOS or iNOS, then you know that what's happening is it's stimulating the synthesis of nitric oxide. Nitric oxide, it's an oxygen. It diffuses freely across cell membranes, but it is very short-lived, so it tends to have a local effect. So it will tend to vasodilate locally, which is a good thing, because if we vasodilated systemically, then our blood pressure would drop into our boots and we'd be in shock. It acts as a homeostatic and inflammatory vasodilator. So it binds to smooth muscle, causes it to relax around the blood vessel. So of course the blood vessel then dilates. As I said, in high systemic levels, of course, if it binds to vascular smooth muscle throughout the body, it's going to drop BP down to your boots and you're going to be in shock. It also reduces platelet aggregation and adhesion. So again, if you have a clot, it not only vasodilates so the blood can get past the clot, it also helps to break down the clot. And also, it's another lysosomal enzyme. Within macrophages and neutrophils, it destroys phagocytosed bacteria. GTN, this is how GTN works. It's converted to nitric oxide to stimulate vasodilation. And also, nitric oxide is the main mediator of penile engorgement and erection. Because, of course, it's a vasodilator. It's a local vasodilator. So that's what's going to happen. Okay, cytokines. Now, this is a specific group, really, They are synthesized and secreted primarily by activated macrophages, other leukocytes including the lymphocytes, but also by endothelial cells. This includes the interleukins. Remember I said there's around 30 interleukins. We're only going to talk about a couple as we go through the rest of the course, really. Interferons that we'll come back to, tumour necrosis factor, usually TNF alpha A, and colony stimulating factors, also known as leukocytosis inducing factors. So interleukins, the main ones that we need to worry about really in terms of inflammation are interleukin 1, interleukin 8 and interleukin 6. And certainly interleukin 1 and interleukin 6 we will come back to when we talk about chronic inflammation in clinical reasoning. Interleukin 10, interestingly, is anti-inflammatory and it inhibits cytokine synthesis. So like the lipoxins in the arachidonic acid cascade, interleukin 10 is anti-inflammatory. Tumour necrosis factor alpha, yeah, we'll come back to this one quite a lot too. It has many pro-inflammatory effects in acute and particularly in chronic inflammation. Interferons, their main role really is antiviral. What they do is a cell becomes infected by a virus, the interferons are released by that cell, go and bind to the cell next one along, the adjacent cell, and cause that cell to start producing antiviral proteins within the cell. So when that cell becomes infected, it's able to resist the virus, it's able to stop the virus from replicating. Unfortunately, it often causes that cell to die as well, but at least the virus cannot replicate. And colony stimulating factors. These are really important at the start of any immune response because what they're doing is they're causing leukocytosis. They are stimulating the release of different leukocytes from the bone marrow. And we have different colony stimulating factors to stimulate different leukocytes. So macrophage colony stimulating factor, as you might suspect, increases the number and activity of monocytes and macrophages. Remember, they're monocytes until they get into the tissues, then they become macrophages. Granulocyte macrophage colony stimulating factor increases the number of all leukocytes, but particularly monocytes and eosinophils. Granulocyte colony stimulating factor is about our first response. So this is probably the colony stimulating factor that will be released first because it causes neutrophilia. It increases the number of circulating neutrophils, and neutrophils are known to be the first responders. As I said, colony stimulating factors, interchangeable term really, are leukocytosis inducing factors. You don't have to remember all the individual ones that I've talked about there, but it's just to help you to understand that there are different types for different cells, and this will determine the phase of the response and what cells are needed now. Also, in terms of the type of pathogen, some of these leukocytes may be more important in different pathogenic infection. Chemokines. Chemokines do chemotaxis. Chemokine, chemotaxis. And what chemotaxis is, it's a release of chemicals that help to guide leukocytes to the target area. So these chemokines are chemicals that diffuse out through the intracellular environment, the interstitial fluid, and are then received by incoming leukocytes, and it tells them which direction they need to go in. So it tells them where to go to find their target. So these are a group of chemical signposts, if you like. Again, a few examples for you, you don't have to remember them, but just to appreciate that different chemokines will attract different cells. So if you have CCR7, that attracts dendritic cells. CCL2 attracts macrophages, CXCL8 neutrophils. There are loads of them. We don't need to worry about the particular names. But if you do see something that looks like that and begins with a C, it's probably a chemokine. There are some other chemicals that act as chemokines, in particular interleukin 8, that has a chemotactic role. So what about our plasma-derived mediators? Let's have a look at those in a bit more detail. So the first one that we talked about was factor 12, Hageman factor. And from this, of course, we can have two pathways. It's an enzyme from the liver. It circulates in an inactive form, and then it can be activated in various ways. So various other mediators can activate it: platelets, endothelial damage, exposed basement membranes. So anywhere where we've lost epithelial tissue and exposed the basement membrane. All of these will activate Hageman factor. One pathway is the kinin system, which results in bradykinin. Bradykinin causes vasodilation and increased vascular permeability. So just like histamine, it's a vasoactive chemical. It also causes the contraction of non-vascular smooth muscle, so it causes bronchoconstriction, and it can stimulate nociceptors. It also stimulates cough receptors. And bradykinin is partly broken down by angiotensin converting enzyme. So in people who have ACE inhibitors, are taking ACE inhibitors, then there is no ACE to break down bradykinin. Bradykinin is uncontrolled. It binds to cough receptors and causes the dry cough that you see in some people who take ACE inhibitors. Our other pathway is the fibrinolysis system. And this, of course, is all about plasmin to break down clots. We also have the reverse of that, the coagulation system, where we have thrombin cleaving the soluble plasma protein fibrinogen to form a form of fibrin that's insoluble. This fibrin that's insoluble then helps to form the blood clot. And last but not least, that brings us on to complement. This is synthesized by the liver as C1. This inactive C1 circulates in the blood plasma, and then it's activated. That activation leads to two sequential pathways. In other words, one pathway leads into another. Apologies for that and for the terminology, but that's the way it is, and you need to recognize these pathways. It's the second pathway that we're really interested in. So we have the classical pathway. So this C1 is activated if it binds to the Fc site of an antigen antibody complex. So remember, the Fc is the bottom of our Y shape, the Fv is the top, the Fv binds to the target. We'll come back to antibodies and their structure later. The Fc, the bottom end of the Y shape, binds to complement, C1. And this activates it as long as that antibody is bound to a target. It can also be activated by CRP. So C1 binds to C-reactive protein, CRP, when it is bound to an antigen. So C1 can bind to either antibodies or to CRP when it's bound to a target, and that activates it. And then we get a whole cascade of enzymatic reactions. As an upshot of that, we get C3. C3 goes into the alternative pathway. So this early part about activation of C1 is the classical pathway, which gives us C3. C3 is at the top, if you like, of the alternative pathway. And this is where it all starts to get a bit more interesting. So, the alternative pathway follows the classical pathway. So, our first one that we're going to talk about is C3b, because we've talked about opsonization. So, C3b will bind to a target and it will mark it for phagocytosis. That C3b, when bound to the target, can then bind to complement receptors on a phagocyte, and the whole thing is phagocytosed. This is opsonization. C3a and C5a, so the two A's, 3a and 5a, are involved in the degranulation of mast cells. So consequently, we're going to get the release of histamine and all those other mediators, heparin, serotonin, etc. C5b to C9 all get together and they form something called membrane attack complex, MAC. Membrane attack complex then makes holes in microbes, in pathogens, or sometimes in host cells if they don't carry a particular protein. And of course, when it forms holes, it causes that pathogen or cell to go into lysis. And this is how it does it. So all of them up to C9, so C5b to C8, attach to the plasma membrane of a pathogen. C9 then joins them and it starts to copy itself. It copies itself over and over until it forms a pore. So in other words, what it's doing is lining, if you like, a hole in the membrane of that pathogen. And just like our cells, what happens is sodium floods in. When sodium floods in, water floods in, and we get cytolysis, or lysis is the shorter term. So our pathogen goes into lysis and bursts. As I said, on healthy host cells, all healthy host cells display a glycoprotein called CD59. There's lots of CD proteins. It stands for cluster of differentiation. There's lots of different ones. CD59, though, inhibits pore formation and it protects the cell from lysis. So healthy cells have CD59, which protects them. Pathogens don't. So they get the full membrane attack complex experience and go into lysis. So that was part one of immunology. We talked about a lot of chemicals and a lot of terminology. And this really is our ingredient list for where we're going next. We need to talk about a few cells as to where those chemicals are all coming from to get that into context. But then we'll look at the processes involved in that phase of innate immunity. What exactly happens and what those ingredients, if you like, are going to make happen in terms of the process as we work through this innate phase. So get that all under your belt if you can before we move on, because it will make the next part much more understandable.